Hi there. So in this video, we are going to look at some of the very famous bioinformatics tools that you can use in your Linux operating system. So this guide is basically an introduction of how to install some of the various bioinformatics tools that include essential for the sequence analysis, sequence alignment, and phylogenetic tree interference. For example, we in the recent video we have used NCBI Blast. NCBI Blast is a suite of tools for sequence similarity searching, crucial for identifying homolog sequences. It is used to search for the sequence in nucleotide and protein databases to find some similar sequencing, aiding in functional prediction of the sequences. So, how did we install the sequence alignment tool, that is NCBI Blast? So first of all, we are going to move to our local home directory, and in the home directory we are going to type in a command sudo. Sudo is actually the permission of your system. Sudo is like the administrative command of your Linux operating system. So after writing the sudo, we are going to write the application name. So application apt slash get is to get the setup of that file, and type install, that means install apt slash get install blast plus, and then type enter. After that it is going to ask for the password, and after that it shows you any error, for example unable to locate the error. So you have to search up for that. For example, we are going to see NCBI Blast plus is the application name. So what we are going to do, we are going to copy the name of the file and then paste it again. Sudo apt NCBI Blast plus is the application name. Now type enter. So NCBI Blast is already at the newest version. So this means it is already installed in my system. So this is the basic command that you have to get. So in the Linux operating system, you have to get the command right and the tool name right as well. For that, I will also be providing my Word file that I made that includes some of the famous bioinformatics tools that you can use, which we are going to see as well. So in this article, you will see some of the basic tools that we need in the bioinformatics degree as well. So I'm going to explain those one by one. So I will open up the slide, and NCBI Blast Plus was the first one. So now you need another tool, which is HMMER. Hammer is used for the searching of databases just like Blast, for the homologs of protein sequencing, enabling the protein family identification. If you have ever come across some of the gene families that you have known, for example, BZIP, transcription factor, IDZ gene family, and some other gene families, you should know this tool. So it is commonly used for the profiles of hidden Markov models, searching, and essential for the functional annotation. So what does it do? So it searches for the hidden Markov model. So how to install this in our system? So again, sudo apt slash get install hammer HMMER HMMER. So it's asking for the permission that after this permission, 7 KB of additional disk space will be used. So that's good for us. Internet is little slow today, so I will not show you the whole installing process, but I will move you to the slides. So JALView is a multiple sequence alignment tool and editor for providing a versatile platform for sequence virtualization. So it is used for visualizing and analyzing multiple sequence alignment, aiding in comprehensive genomic and evolutionary analysis. So you can read the tool on the internet as well, but this is a basic introduction that I can give you. So there is another tool for de novo assembly software of the short reads and genome, which is called EBIAS. It is used for the transcriptome assembly as well. It is used to assemble the short reads into long contigs or scaffold without a reference genome, critical for the species without complete genome identification. So you can install this tool by the command which is given below. Gmol is a molecular viewer and editor offering interactive 3D representation of biomolecular structures. You can use this for visualizing and analyzing molecular structure at atomic level, facilitating the protein modeling and structure validation. You can use this tool by typing the command sudo apt get install gmol. So we move on to the sequence alignment section. MEFIT is a tool that is used for the multiple sequence alignment for its high accuracy and efficiency. Muscle is also the robust multi-sequence tool which is used for the large-scale sequence analysis. For example, if you have large dataset, you can use Muffle, but if you want accuracy, you can use Muffet. Cluster Omega is state-of-the-art multiple sequence alignment tool designed for the higher accuracy alignment. It is used for accurate alignment of multiple sequencing, ensuring reliable evolutionary and functional analysis. We will also use this tool. So this is the tool that you can also use in the sequence multiple sequence alignment analysis. There are also some tools used for the phylogenetic analysis. For example, Mr. Bayes is the Bayesian inference of phylogeny construction. You can use this tool by installing it. NJPlot is the visualization of phylogenetic trees in various formats. Then comes the phylogenetic analysis tool for the tree construction of maximum likelihood estimation. These are some of the terms from the phylogenetic. If you want to learn more about them, you can Google it. These are some of the common terms that are used in phylogenetic tree interference. So you can use these tools for this analysis. Then there comes the protein structure visualization. You can use PyMol for that. Data visualization and analysis of protein structure can be done with PyMol. Then there comes the JMol, interactive 3D representation of biomolecules. You can use JMol for that. It is a very good tool. You can use both of these for the sequence analysis. So let's just say we want to install any other tool, so you can use this command. Then there is a Word file that I have provided for you guys. Here you can use this to install all the tools that I have given. For example, you can install BEMTools and RASMol and all of these to install according to your own needs. So that's it for this lecture, and in this lecture we have learned how to install some of the basic bioinformatics tools, and it is still installing because the internet is quite slow today. So that's it for this lecture, and I think this is enough for how to install any package. So in the next lecture we are going to use one of these tools to perform any analysis. So that's it for today.